Dumelang Abshani, hello. You are tuned into TMZ on a mission up with SABC 1 Mzansi for sure.
Yeah.
It brings you nothing but the best inside tech edutainment.
Hosted by your favorites. I go by the name of Caddy, and I'm coming you one buffet to this is Ubuntu Wabuntuano. I'm talking about buffet to now going to be all about metagenomics. So SABC deep down into the tiny dot of your DNA. I know it sounds like a tongue twister about metagenomics.
Yeah.
But buffet by the end of the show,
yes, let's get straight into it, buffet. What is this episode about?
Go.
Sheba fela re le tshwaretse mo lenanehong la kajeko la TMZ. Kajeko le here batla fumana tsebo e ngata in the world of bacteria through metagenomics. Ebile here batla bona re hantle the metagenomics le soil metagenomics di sebetsa jwang. So yona e batla a role e bohlokwa haholo ka mahara farm ebile kgona ho re fa our food source. Re batla ho ithuta ka science ene ka mamra metagenomics re bona re hantle e kgona ho thusa jwang on how sustainability is linked with metagenomics, specifically soil metagenomics. Re batla ithuta ka the origin of metagenomics re hantle ho tlile jwang hore e be teng. Re bona re hantle he ba ifumane jwang. Re bona re ke mang o hlale feeling haholo re testele di memory skills re bona ke mang the big buzzer braids ha re bapala the TOMZ quiz.
Alright, but I didn't tell you something ne. Without you guys, we'd literally be a bunch of people talking to themselves. So you guys are pretty important when it comes to teenagers on a mission coming into fruition, which is why we're asking you guys a question on our social media pages, and our question is as follows. What is biochemistry? Sipeteke u-amp yena uti it's the study of chemical processes. Sipeteke notemba uti the branch of science concerned with the chemical and physiochemical processes as well as substances that occur within living organisms. Ay ingati natural science why dweba oputiza. Please do keep those comments coming. We're gonna go to our school and see what the kids there have to say. Masamen.
Metagenomics is what we are talking about. Namhlanje bafethu sesishayile touchdown yethu sise Nirvana Secondary High, and right next to me is a young gentleman. Esimbuza ama questions bona ukuthi uzokhona na ukuwaphendula. Putam, how you doing?
I'm good, and you?
I'm great, man. What's your name?
Ritabile Shok.
Ritabile. Can you tell me what is metagenomics?
Is the study of growing plants, for an example, is things that we cannot see with our naked eyes, which is living bacteria, DNA. Yeah.
We ask that pollution plays a major role when it comes to like destroying our environment. Ne? How does pollution interfere with our soil or interrupt our soil?
It makes the organisms within a plant, actually the plant just dies.
Do you think plants have DNA?
Well, no, they don't have DNA.
Are you sure?
Yes, they have RNA.
Okay.
The living soil. Re batla bona re hantle clean microbes tse sa bonahaleng ka mahlo di kgona ho affecta jwang dijo tse re di jang. Metagenomics ke a study of genetic material e leng re kgona ho fumana ka mara environment ya rona ka di samples. Genomics yona ke a useful field of biology e leng re focus on building, purpose, growth planning and editing of genomes. Genome is an organism with a complete set of DNA including its genes. The scientists study bacteria le fungi to relate to the plant in ways that can make the plant grow better. Knowledge an increased crop making for expanding population. Dip yourself in the soil and find out how plants are helped and hurt. Some microbes interact with the roots. An increased crop making for expanding population.
Who knew testing is something was so important? I mean, it helps us understand the lifespan of plants, right? It helps us understand the evolution of the earth, as well as fungus and how they react or interact with other plants.
Yes, sir. Asbonoguti, himapi ama young scientists that are walking the soil of this earth to try and change the world. Check this out.
Let's see.
My name is Juanita. I'm in grade 12, 17 years old at Creston College. One of the major problems that we are facing today is that we are having a global energy crisis. Despite being so advanced technologically, many people in South African rural areas still do not have access to electricity. It is a problem because many people now do not have access to the internet and many other technologies that are available through the means of electricity. The solution I found was a series of instep soles that one could place into their shoe, and as they walk, the pressure of their feet against the ground would result in an electric charge, that could then be used to charge a battery. This technology is called piezoelectricity, which is a conversion of mechanical into electrical energy. Without even realizing it, we use this technology every day, such as in our speakers and microphones on our mobile devices, as well as many other uses. An increased availability of electricity would result in a great improvement for all, especially the learners that would now be given opportunities to access the internet, as well as other learning resources.
Ka the late 17th century, Anton van Leeuwenhoek, kena olengompili, olengafumana metagenomics, directly studied organisms selling or held for manikamara a pond, likamara menu ahai. Ka 1920, cell culture, kiona elenga change to RNA sequence, eling or improve bacteria. Ka the 1980s, unali disagreements, seveling badi fumana. The cells that are workable but not culturable, and Norman Peck, eling apena an idea, eling or copper DNA, eling or takamara environmental nakadi samples, ka 1985. Report ya pele ele mai publisher ki Pace Lady colleagues if it ka 1991 ele ngoro ba reportile the useful genes. Libitele bitang hore ki Metagenomics ke ntho ele ngoro ba sebedisitse ke Joe Handelsman, John Clardy, Robert N. Goodman, Sean F. Brady le batho ba bang ba lenga ba fitla ka publication ya teng ka 1998.
Biochemistry is nikeza benefits ezininzi within the world of science, as well as fields that have nothing to do with science.
That's why my scientists bafethu kena bona they're on a mission to find out exactly ukuthi kenzakalani le emhlabeni nokuthi iziphi izinto kena that can make this world a better place, and mapha ama solutions akhona bafethu.
Yes, to epidemics such as pollution, for example. Funeka sikhumbule bethunana that yonke into apha kweli lizwe iyasebenzisana, both negatively and positively.
Yes, all we have to do is hold hands and make the world a better place. We'll see you after the ad break. We're back with Faith Session. We're looking to supplement to our channel, your #1 channel, SABC YouTube for sure. Today we're all about biochemistry. Sifunda kabanzi about metagenomics. Uthi sinqoda kanjani empilweni zethu ukuthi ama scientists asebenzisa kanjani Liana in metagenomics to actually figure out what's happening in the world and which soil is great and which soil is not. But esikhathini samanje sonikaya did you knows.
Beniyazi ukuthi biochemistry is quite important in the development of scientific disciplines, including forensics, botany, molecular biology, the list goes on.
And when it comes to biochemistry, the word derives from a combination of bio, meaning impilo, and then chemistry.
Yes, and when we think about it, biochemistry has been an ancient concept with the Chinese basing their formula on the formation of yin and yang.
With that being said, bafethu, sihamba sovakashela our expert wanamhlanje usichazela kabanzi ukuthi kahle kahle basithi metagenomics. What is that? What's up? What's going on, Bafethu? It's your boy C-A-W-D-Y, pronounced Caddy. Yet again on another mission, and this time we're running the streets of Pretoria. Since we touched down to CSIR and Duana. So I'm sure we're all about metagenomics. What is that? And it contributes to impulse. Without wasting any time, let's go meet our guest. Let's go. Sabon.
Hi, good.
Unjani?
Ngiyaphila.
Ngiyaphila. Namhlanje sokuvakashela and welcome to the show.
Thank you for having me.
Yes, so namhlanje sifuna ukuthola kabanzi about molecular biology. So sikhuluma nge-metagenomics, ne?
Metagenomics.
Yes, metagenomics. There we go. But before we get into that, what do you do and where are we?
Okay, igama lami Nanusibongile. I'm from a small town in the Eastern Cape. Right now we're at the CSIR, the Biosciences. What I do now is recombinant protein production.
Okay.
So I'm in a biochemistry lab.
Okay.
Yeah.
So sisiwamsichazisele kabanzi ke ngokuthi umsebenzi wakho kahle kahle uyenzani?
So my work entails metagenomics, like you had said.
Yes.
So in metagenomics, it's actually a study of whole genomic DNA from an environmental sample.
Yeah.
So we isolate DNA from a soil sample, in our case, which we can then use further with the steps that are involved in metagenomics for recombinant protein production. So we target different enzymes, different proteins, depending on what we are looking for at that particular moment.
Yeah.
Yes.
And niyenza lezi zinto lezo ukuthi you guys extract DNAs from samples.
From the soil samples. Okay.
Yes.
So the reason why we use metagenomics is that most organisms within some environments are unculturable, so you can't detect them.
Okay.
So then when you use metagenomics, you can extract a whole range. You can cover the undiscovered area within that sample, and you can get those DNA, which can then be readily used for genes that can be expressed.
Yeah.
Yes.
Okay.
So, after transformation, where you now have it, you'll take your plate, you scrape off a single colony, then you grow it in a different medium, which is a liquid medium, where you will now start the process of expressing your protein. So you take this, you put it into an incubator after you inoculate it, you grow it until for 16 hours. Then after that, from that, you'll take a fraction and transfer into a fresh media. The same thing.
Okay.
Then you grow it until the cells reach a certain optical density. You measure that using a specialized equipment as well. So you don't just look at it and like, okay, it's fine now. You measure. Then once it reaches that optical density that you want specifically, you then induce the cells. Then they start making more of the protein that you want. Then you take this, you transfer into a falcon tube, which is this. You transfer it in here. And then you use a centrifuge to spin it down so that you can separate the cells.
Okay.
And then you'd have a crude sample, crude fraction. So once you spin down, you are most likely to get this to be your crude. So your crude in this case means that it's what you want and other proteins that are expressed within that cell. So now you have to purify it to clean it out and have your protein of interest. Then we use specialized columns. So now this is for a small scale. So most of our proteins are tagged. So you add a tag to it that will help it bind to this membrane. So this is a purification column. It's an IMAC. It uses ion exchange. So it's like charges. Then you take your crude, you use gravity flow. So you take in 3 mils, you allow it to flow through, you collect all the fractions, and then you wash it out using another buffer, which is another solution. You wash out the ones that you don't want, with yours having been bound to the membrane here. So a protein gel, we then use, we cast it using bio-red products. So you just mix the solutions using the owner's manual. And then you go load. So before you load your gels, once you have your fractions, you have to denature them. So you have to add a tracking dye, which is called an SS dye. You have to denature, and then you put it at 95 degrees, you heat it up. Then you can go load your samples on a SCS gel.
So are they loaded now?
We can go load them.
Let's go.
You can do the honors of loading the gel.
Let's do it.
All right. So now we're gonna load this and start running it. Yeah. So I'll load one, and then you can
I'll load the rest.
have a try. Yeah.
It doesn't dissolve, really. It stays there.
No, it will stay there, yes. So the gel is made into 2 parts. There's the stacking part where you do the loading, and then there's the resolve part where it migrates, and then it separates on the sides. So you get your tip.
So I just...
yes. Then you push it to the first stop.
Then I let go.
Release this, yes. There you go. Then you load it on number 3. So you load the samples based on the fractions. You need to start with your crude, the flow through, which is the crude after it went past the column. Then your wash, where you've washed out the protein, and then your eluted fraction. Then you have to run it. You have to make sure it's negative and positive, negative, positive.
Yes.
And then you set it to run. It will run roughly for 2 hours until it's done. Then when it's done, we deassemble, get the gel, stain it using a composite. It's a blue stain.
Okay.
We stain it, then the entire gel will be blue.
Okay.
Then you get a destaining solution, which will remove the blue from the rest of the gel.
Okay.
But all the protein bands will be stained, so they won't, they will remain on the gel. Then you can visually see the whole process when it's done. So that would have been your marker, your crude, your flow through, your wash, and your eluted protein. So you get that specific band, which means now with the protein that you have, that you eluted, you can start your process of characterizing it based on its function. So if this was, let's say you were expressing a ligase protein, then you can start doing some characterization in the lab where you take broken DNA molecules and you use the enzyme with buffers and religate them to circular DNA once again.
Oh, okay. Susan, thank you so much for your time. I've learned so much. Guys, right about now, I'm still going to be a scientist in Zabing Plumelana. But for you guys, go back to studio. Mission to Pilelalana. I'll see you guys next time. Adios!
With the assistance of biochemistry and metagenomics, the world is becoming a more effective and simpler place, just in terms of farming and general plant life.
Yes, so mine don't come did you know is buffet. So did you know with metagenomics buffet is important because of the range of bacteria in kubalulekile for the soil health and the human health.
Definitely. And when it comes to the employment of biophysicists as well as biochemists, this is projected to grow 11% from 2016 to 2026. This is larger than the average growth of all occupations.
Okay, when it comes to UCT, Stellenbosch, those are the best universities when it comes to biology and biochemistry, because when it comes to research, bapuma phambili, and they have top notch when it comes to knowledge.
I can really see that, people in the lab doing their thing. All right, let's take a quick ad break. So we're going to
Let's go.
Yes, Katie. Ngoku ndikhulayo ne ndiyaqonda uba science is quite important in planting, yada yada yada, water, sun, yes, photosynthesis. Kodwa I didn't know that science could take it to these levels.
It's crazy, ne?
Wow.
Sifunda kakhulu la ku-teenagers on a mission. Bafethu khona manje sibona ukuthi kahle kahle esigela ubani ocavile owazi ukuthi zikhiphani when it comes to the world of metagenomics. Let's go have a look. Hello guys. How are you guys doing? Okay, cool. My name is Caddy. I'm from a show called TOMz. You guys know TOMz? Yes. What does TOMz stand for? You guys know the show. Bathi wayisusa uzoyimela thina siyisusile ke ntwana. Welcome to it. Lena ke sibiza nge-TOMz Quiz game show ntwana. Niyazi ukuthi sidlala kanjani le games. Vakashela abangani bethu eSkela, play with them and see kahle kahle ukuthi who will reign supreme. We are in the building, the gents are in the building, the girls are in the building. Sifuna ukuthola kabanzi ukuthi kahle kahle what's the name of the school and where is the school? Where are we?
Yeah, in Nirvana and Malaysia.
What grade are you guys doing?
Grade 11.
Grade 11. See, Shiley touchdown here to Bafethu. Nkumulo ukuthi nani lapha emakhaya. You can always play with us. Pretty simple. Go on our social media pages. On Facebook, ngiyankhumbuza Bafethu, we are TOMZ. And on Twitter, we are TOMz_SABC_10. Can you guys please make some noise? Okay, cool, guys. So this is how we play the game. You got a buzzer in front of you, ne? I'm gonna ask you guys questions, ne? My questions, guys. I'm gonna give you multiple choice from A till D, ne? After reading the multiple choice, then you guys can press the buzzer. You press the buzzer when you know the answer, ne? If you don't know the answer, don't press the buzzer and then you think about the answer, ne, guys? You guys got the rules? Are you guys ready to play? Sure. Okay. First question, please. Question who carries us as babies in their tummies? Is it A, our friendships, abangani bethu? Is it B, our mothers? C, our fathers? Or D, our brothers? Girls, you guys press first. Which one is it?
B.
B, indeed, our mothers. Give it up for the girls. Our second question is, where do we get our microbiomes and DNA from? Is it our grandparents, A? B, our parents? C, our immediate family? Or D, all of the above? Which one is it? Gents?
All of the above.
And the correct answer for the second question is definitely D. Give it up for the chance. Third question is, can bacteria be found in soil? This is true or false? Girls.
It's A.
Let's find out whether this is true or not, and it's definitely true. Hey, give it up for the girls. Moving on to the fourth question. As the fourth question here to Iti. Is all bacteria bad for a person? Is this true or false? Gentlemen?
B, false.
And the correct answer is definitely B, false. Give it up for the chance. The last question is, can better soil help grow better food? Is this true or false? Okay, girls.
It's true.
And the correct answer is definitely A. Give it up for the girls. Okay, cool, guys. This is how we ended off. The girls take this round. They are our big buzzer brains. Until next time, keep it fresh and keep it training. Don't forget to also hashtag TOMZ Quiz Game Show on Twitter and on Facebook. Bye-bye.
Yabo basifike ekazini ntwana zikhona lezi shango triple P yazi triple P umele
Halala.
yazi umele
Yeah.
awungitshele lapho bakhumbuze
Big buzzer brain. Big buzzer brain.
Shout out to you Bafethu for playing with us. You guys are amazing. Laba baluzile ngawari Bafethu nani one day nizoyithola triple P.
Okay, let's check out what the future holds. Since we have big buzzer brains, I'm sure you'll be able to handle this one. Masame.
The future of biochemistry, Metagenomics, is science of the future. Biochemistry is fast developing into a very important subject. It's a groundwork research biochemistry, Metagenomics, study setting is a successful career offering. Request biochemistry meaning the fields of medicine, pharmaceuticals, biotechnology, microbiology, veterinary, agricultural, and dairy sciences. Biochemistry has the benefits the fields they affect the human race, the ecosystem.
Alright, Miranda, we gave you an incredible beginning, a spectacular middle, and this is the end. So let's leave it on a high note, Miranda. We have learned a lot. Timugutu, you guys will write notes down, share it with your teacher, show them that you do some homework on the side.
If you missed the episode, we've got to like the TV Ntwana. Hamba ku www.sabceducation.co.za/TOMZ to get more information. Or you can simply follow us on our social media pages. On Facebook, see TOMZ. And on Twitter, see TOMZ_SABC_10. I'm Fresh by Katie on Twitter.
Abunda_P on Instagram. And goodie bye.
TOMZ. A materials engineer and 3D printing expert introducer to the science that welcomes us into the incredible local laboratory yako CSIR. A sneak peek into the exciting innovations 3D printing for a sustainable future. TOMZ 2 SABC 1 Mzansi for show.